So you're going to hear today about how life is strange by learning about three peculiar, legendary heroes of survival. We have three great speakers for you today. Taryn Olivas is in the Department of Cell Biology at Yale University. She studies organelle growth during autophagy, and she started her career in science by tutoring chemistry students in undergrad. Yangqi Gu is in the Department of Molecular Biophysics and Biochemistry at Yale University, where he studies electrogenic soil bacteria. Outside the lab, he loves to cook and apply the skills he's learned in science to the kitchen. And finally, Carrie Ann is in the Department of Genetics at Yale University, where she studies how neurons respond to injury in nematodes. Her fun fact is that outside the lab, she also enjoys swimming, running, and playing classical piano. These three speakers have a great talk planned for all of you today, and that talk focuses on tiny little heroes. In difficult times, we often look to heroes. We look to real heroes who will guide us or protect us and those we care about. And we look to imaginary heroes that inspire us and let us escape for a minute or two from our current situation. Today at Science in the News, we give you a little bit of both by taking a look at three legendary survivors that you might never have expected inside our bodies and in our own backyards. First, we'll hear from Taryn, who will tell us about the Autophagosome, a strange cellular hero that keeps our bodies alive. Then we'll hear from Yangqi, who will tell us about a special bacterium that we can use to generate electricity. And finally, we'll hear from Carrie Ann, who will tell us about how a peculiar little animal might one day help us transport vaccines. These three little creatures are small and strange, but in their own way, by doing their part to protect us and teaching us how to improve our societies, they're more heroic than we might think. And today, you're going to hear their stories. So, without further ado, I'll pass the screen off to Taryn, who's going to kick us off with the Autophagosome. Take it away, Taryn.
To unshare your screen.
Yes, let's see. How do I do that? Here we go. Faces were covering it. All right, all good, Taryn.
All right. Can everybody hear me? Okay. Thank you so much, Caleb, for that lovely introduction, and I'm very happy to be kicking us off today with the first talk. Today I'm going to tell you about the protective role of autophagy in cell survival. The background here is an artistic rendition of the inside of a cell, and that's because with this first talk, we will begin at the smallest scale of hero, a hero that works inside a single cell during the process of autophagy. Autophagy literally means auto, self, phagy, eating. Therefore, autophagy is the process of a cell eating itself. This, of course, prompts the question right off the bat. Why in the world would a cell want to do that? It may be strange to think about a cell eating itself for any reason. You might be thinking that eating oneself is even counterproductive to survival, but the key is in what is being eaten. The typical everyday function of autophagy is in waste disposal. That's right, autophagy is really a process of garbage collecting inside the cell. Things inside the cell become broken and unusable over time, and you can imagine that a buildup of trash would prevent the rest of the stuff inside the cell from functioning, and eventually even kill the cell. Autophagy protects the cell from this fate by cleaning up the trash in the cell before it causes problems. A clean cell is a happy and healthy cell. Our bodies are made up of cells. These so-called building blocks of life make up each and every organ and tissue in our bodies. They have all sorts of shapes and sizes and functions that allow them to do their job to make you, you. But no matter what they look like or do in the body, they all have autophagy in common. No matter the cell type, if anything is damaged inside the cell, it gets in the way of other things in the cell just trying to do their jobs. To get rid of what is now garbage, there's essentially a game of Pac-Man going on inside every cell, regardless of what type of cell it is. Who here has heard of Pac-Man? For those of you who haven't, and if you haven't, you should definitely try it. Pac-Man is an old video game with a simple premise. You are this little mouth that tries to eat all of the white dots on the board. There are enemy ghosts that can kill you, but there are also these bigger white dots that make the ghosts vulnerable to you eating them. There's also fruit that pops up here and there for bonus points. Now, if you're all ready, I'm going to tell you about Pac-Man in the context of biology. Inside the cell, this Pac-Man-like process is one of housekeeping. Imagine that instead of food, Pac-Man is collecting garbage inside the cell. Broken things and stuff the cell doesn't need anymore. Pac-Man is executing autophagy by collecting trash inside the cell. To explain how autophagy is carried out, it's time to introduce the Pac-Man of the cell. Remember that autophagy literally means self-eating. In order to carry out this self-eating function, the cell forms a structure called the autophagosome that grows around garbage inside the cell. You can see the resemblance between the autophagosome and Pac-Man already. The autophagosome is the hero our cells need to keep them alive. It's important to note here that unlike the game, inside the cell there are multiple autophagosomes acting during autophagy. It's more like multiple Pac-Men working to clear the board, each only taking one large gulp. When I refer to the autophagosome, it's standing in for a collection of autophagosomes. In fact, responding to severe stress, the cell can generate more than 100 autophagosomes to spread them out for lots of self-eating at once. On an individual level, each autophagosome will grow around trash in the cell in a process similar to Pac-Man closing his mouth. Once closed, the autophagosome cannot digest the material on its own, so here, Pac-Man gets a power-up. A structure called the lysosome will fuse with the autophagosome to become a structure called the autolysosome, one large structure that is now capable of digesting the trash. You can think about it like Pac-Man becoming larger and more powerful. Indeed, this is how it expands inside the cell to capture garbage. I want you all to see it as it is in the cell, a three-dimensional bubble containing anything it formed around that merges to another three-dimensional structure, the lysosome. This fusion step is very important to complete the process. In order to clear the cell of all this garbage, Pac-Man is going to need the help of the lysosome. The lysosome is commonly referred to as the garbage can of the cell. I like to think about it more as a recycling bin, as components of trash can be used again by the cell. If you were to cut the lysosome open, inside you would find special components that break down any trash that goes inside it. You can think about it like a stomach, a walled off structure that's full of acid that breaks down the food that we eat. The components inside the lysosome work like stomach acid to digest cellular garbage. So if the lysosome is the recycling bin, then the autophagosome is the bag that has collected the trash that goes inside. After Pac-Man fills up on trash in the cell, he'll do a majestic alley-oop into the recycling bin. The lysosome is the end of the autophagy pathway, just like receiving your new high score is the end of your round of Pac-Man. The main reason we need autophagy is because the autophagosome is particularly good at consuming large quantities of trash and large items. But aside from its life-saving ability to pick up trash in the cell, it also uses these properties to protect the cell during various stresses. Two more examples of survival functions of autophagy are its response to starvation, in which the autophagosome eats random, unimportant things inside the cell, and its response to consume foreign pathogens, that being any bacteria, virus, or microorganism that can get you sick. I'm going to expand on these functions for you today, beginning with the starvation response. Autophagy protects the cell by eating random things inside of it when it's starving. This protective role is what gave the process its name, which you will recall means self-eating. We all know that we need to eat. Nutrients that we get from our food become energy for our bodies. When we don't get these nutrients, we begin to run low on energy, and this is essentially what starvation is, not having the fuel needed to keep the body running. It's a good thing that our bodies prepare for this. Our bodies prepare for starvation by storing the excess of what we eat as fat. I know most of us aren't thrilled with this fat storage, but it is a rainy day fund of nutrients for the body during starvation. Between the fat and the muscle on our bodies, there are enough nutrient reserves to survive starvation for quite a while. In other words, your body can eat parts of itself to survive, just like the cell can eat parts of itself. Faced with persistent starvation, each individual cell will consume unimportant parts of itself to stay alive. This is different from storing nutrients for later. The cell must prioritize survival temporarily and eat anything inside itself that it does not immediately need. This will provide it with energy until it receives nutrients again. If we look inside the cell, there are multiple autophagosomes inside the cell, and will represent the unimportant cell components as things that Pac-Man likes to eat. The autophagosome will close around the components, carry them to the lysosome, and fuse with the lysosome. Now the trash and the stuff inside the lysosome that can break down that trash are mixed together. Once the process is completed, the lysosomes are recycled and the broken down cellular components can be used for energy during starvation. This really is a quote-unquote green process, since all of the digested components will be used to feed the cell, and even the lysosomes can be reused. During starvation, our hero, the autophagosome, rescues the cell from certain death. This is just one of the amazing ways the autophagosome protects the cell. Now that you've seen what happens in the cell when it's starving, let's look at the autophagy response to invading pathogens, the third and final protective role of autophagy that we'll discuss today. For this defensive role, we will explore what happens when the autophagosome alerts the cell to danger. As I said in the beginning of the talk, that large dot in the game allows Pac-Man to eat the enemy ghosts, so we'll treat it as the alarm signal. When a virus enters a cell, it begins to make more of itself. Each piece is assembled within the cell using the cell's own machinery against its will. While the autophagosome is carrying out its natural trash collecting function, it may run across and capture a piece of the assembling virus. It will then alert the cell to the presence of that virus. This alert is just like the large dot in Pac-Man that makes the enemies afraid. Once the cell is alerted, it will know it has been infected and begin the immune response to combat the invading pathogens. This will effectively mark the invaders for digestion, and they should indeed be afraid, as our hero, the autophagosome, has blown their cover. Now Pac-Man can eat the invaders and help the cell protect itself. While the immune response is many complex actions taken by a cell and by the entire body during an infection, autophagy can do its part from within a single cell to capture and remove pathogens. I'm about to show you an incredible video of an autophagosome growing around targeted bacteria. The green is the autophagosome, and the purple is invading Salmonella bacteria. You may know Salmonella as the bacteria from raw chicken that can give you food poisoning. As we watch, remember that the autophagosome capturing a bacterium can protect you from getting sick. Here, if you look closely, you can see the autophagosome growing around the Salmonella bacteria in a process that takes around 10 minutes. Again, the green is the autophagosome and the purple is the bacteria. The autophagosome grows around the bacteria, and this is what I'm referring to when I say the autophagosome eats trash or invaders. It's pretty cool. Once engulfed by the autophagosome, the bacteria can be destroyed inside the lysosome. When the cell doesn't feel well, and we look inside of it, we see autophagosomes capturing enemy pathogens just like Pac-Man eating the ghosts. Importantly, the same process for bringing the pathogen to the lysosome and fusing with the lysosome occurs in this process just as it does during starvation. This is the way in which the autophagosome always destroys its contents and defends the cell. Now, while the natural roles of autophagy and cell survival are amazing on their own, scientists are now asking if we can harness autophagy to make it defend against what we want it to specifically. The question is, can we determine what Pac-Man eats and when? One condition scientists hope to treat with autophagy that I will talk about briefly today is neurodegeneration. Neurodegeneration is the breakdown of the brain that results in issues in a person's movement and or their mental capabilities. The neurodegenerative disease that I will focus on here today is Alzheimer's disease, the one you probably all know best for its heartbreaking effect of erasing memories. In this disease, the brain cells get bogged down with tangled proteins and dead cell components, separate symptoms of this disease. The reason scientists are thinking autophagy could be helpful is due to its naturally high efficiency inside brain cells. You may recognize this cell from the beginning of my talk. It looks so different because it has a unique function in the brain. It is because these cells do not divide like other cells that they need autophagy to clean them up more often. Tangled proteins and dead cell components are normally trash the autophagosome should be able to clear, but in the later stages of the disease, it's unable to close, like Pac-Man with a stick in his mouth, holding it open and rendering him useless. Preliminary preclinical studies in animals and cells are working to use autophagy to clear the trash that clogs up brain cells in this disease. In the early stages, when autophagy is still working, the focus is to increase autophagy to clear as much as it can. In the later stage, when autophagy is stuck before the lysosome step, the focus is on increasing the efficiency of autophagosome-lysosome fusion so that autophagy can progress normally again. This will include making sure the autophagosome is closing and making sure fusion can occur. It may go without saying, but the more scientists learn about how the autophagosome forms and works inside the cell, the better chance they have of generating therapies using autophagy. This is one reason why people like me and my lab are studying how the autophagosome forms. So what have we learned today? We've seen that the autophagosome is a cellular hero that feeds the cell in times of starvation, is capable of capturing invading pathogens, and cleans up the unnecessary and broken components, the cellular garbage, and takes it out. But most importantly, we've learned that the autophagosome is the defender of the cell. It is the hero that our cells need. All right, thank you for listening to me today. Next, you're going to hear from my colleague Yang-Chi, and he's going to increase the scale of hero from within a cell to a whole bacterial cell. He's going to reveal another amazing survivalist, a bacterium that the autophagosome would not want to eat.
All right. Thank you so much, Taryn. Hello, everyone. I'm the second speaker today, Yang-Chi. I first like to show a huge gratitude like you can all make today. As my colleague Taryn has presented this beautiful but strange micro-scale surviving strategy inside a cell, now our journey of strange life will continue with the larger scale to look at individual cells, particularly bacteria, and see how their surviving strategy could generate electricity. Sounds already strange, right? But in order to see how this strange thing could happen, we need to understand just 4 simple elements: bacteria, breathing, electricity, and electrons. So the first thing is bacteria. These are the tiny organisms that will be the focus of my talk today. And the second is breathing, a process during metabolism to convert food into the energy we need. Third is electricity. This is the stuff powers most of our machines around the world. And the last element is electrons. Those are the small particles, so small that invisible to our human eyes, but they're responsible for the world's electricity. So later on, I'm gonna explain each of these elements in details. But as we can all see here, these 4 elements are closely related, and if they can work together correctly, they can teach us how to use bacteria to power our society. So first, I would like to ask all the audiences one question. How do we feel about bacteria? Pathogens, germs, those little creatures give us cooties. Those are the common terms associated with bacteria on top of our head. However, bacteria have survived more than 3 billion years, much longer than the existence of our human beings. Thus, as a biologist, I would like to justify their name today by showing you how these natural great survivors are not bad guys, but actually heroes who use their superpowers to help us and support our lives. So their superpower comes from special ways to breathe. We all know how to breathe. Breathing is the simple motion we do every moment of our life. It's the only secret to longevity and the only power to keep our loved ones alive. Like our humans, all animals and plants know how to breathe. But unlike our humans, some bacteria, they breathe in a rather strange way. So all our human beings, all the animals and plants, they breathe by consuming oxygen. Every breath we take is going to deliver the oxygens into our body, to the cells, and while taking out the waste gas, so the food we eat can be converted into the energy we need. However, for some bacteria, they breathe without using oxygen, and they will still be able to convert the food or the nutrient they need into their survival energy. But I want to point out that they share the common feature applied to our breathing strategy is that we both produce these small charged particles called electrons and direct them towards a certain direction for the source to receive them. In the case of our human beings, the electrons are directed to oxygen, while in the case for some bacteria, the electrons can be directed to somewhere else, which can be further made into electricity. So what are electrons? Electrons are the particles so small that are invisible to our human eyes and even smaller than atoms. They are negatively charged, as I showed you in the schematic with the blue balls and a negative sign here. They are the particles generate electricity to power our society. However, under most conditions, these electrons are randomly moving around and unable to generate electricity like undisciplined children. However, also as kids, if a proper guidance was given, these electrons can be directed into order and which can further flow in sequence, just like how the water running down the river. When electrons flow, they become electric current, and which can further generate electricity. Depending on the scale of the flow, they can manifest as small, as harmless as a zap, or as dramatic as a thunderstorm. So as I mentioned before, the process of breathing also directs electrons, especially for those bacteria which do not use oxygen to breathe. their electrons can be directed to somewhere else. So if we guide these electrons and connect them to any external storage to receive them and complete the flow, we can generate electricity. So why do we care about electricity? Well, ever since the 20th century, we have entered the age of electrical power. Electricity not only illuminates us, but also connects us with the rest of the world. Actually, at this right moment, we are also borrowing the power from electricity. It has greatly shaped our ways of living and improved our life quality. But as almost every convenience comes with a cost, electricity comes with its own issues. So the problems not only lie in money, but also put a huge burden on our environment and also limited resources. The major issue comes from the way we generate electricity. Traditional ways to generate electricity have to consume a lot of resources and cause severe air contamination, producing hazardous waste, or invading natural habitats for other species. So we need to look into alternatives alongside with some newly developed energy, such as nuclear power or solar power, to alleviate the problem. So as a scientist, I want to ask, what would be the alternative way to make electricity? The solution has been passed down by nature over generations. We can take advantage of the breathing power of this special bacteria to generate electricity. So who will be our heroes today to provide the answer? Here comes in my personal hero, Geobacter. As the name Geo suggested, they are the bacteria living in the soil, covering over 90% of our Earth's surface. The first species of Geobacter was discovered from a ditch sediment from Norman, Oklahoma, by Dr. Derek Lovely in 1987. And sequentially, over 20 species of Geobacter have been discovered. And here I'm showing you a representative picture of the Geobacter with this elongated cell shape and their raw cell shape. So I want to point out that these bacteria are the bacteria which do not use oxygen to breathe. Also, they're environmentally beneficial bacteria, meaning they're able to decompose many heavy metals or waste, which could be a good helper cleaning up sewage water or our environmental contamination. They're not only environmentally friendly, they're also the natural factory of electricity. As I mentioned before, we need to connect the breathing object to direct the flow of electrons in order to harvest electricity. These bacteria are doing us a huge favor by growing these cables, which help us connect them to any external source. They produce these protein filaments, which we call Geobacter cables. They act like the same cable we use every day in our daily life, which connect our TV, turn on the lights, or charge our phones. So we can simply plug these bacteria to an external storage and direct these flows of electrons generated during the bacteria breathing. I also want to point out the beauty of this process is that electrons are the byproduct of the bacterial breathing. Thus, by removing the electrons from the cell body, will not harm the cell, but rather helping them with their metabolism, which means we get free electricity by helping each other out. But as a scientist, we don't want to stop here. In order to optimize the process and increase the yield of electrical power, we apply various tools to study them and understand the process in depth. We can use biological tools to visualize the protein, the Geobacter cables, study their components, figure out what they're made of, or how they look. But we also borrow many tools from physics or engineering, similar to a tool, let's say multimeter, to study how good the performance of the Geobacter cables are. So the multimeter is the tool commonly used by electricians to monitor the flow of electrons, which can further tell you the performance of your electronic devices. Thus, we're applying a similar tool to tell how good our Geobacter cables are. So we want to achieve the goal I first proposed, to generate green new energy. These bacteria have provided us with a blueprint to generate green new energy. Their surviving tips have left us with a great fortune, and in the near future, we can dream of living in a house powered by these bacteria. But of course, we cannot live on a far-fetched fantasy. We need to put them into reality. Here are the two examples I'm showing you here to demonstrate how close we are from the future. The first example is the research I'm actively participating to engineer better protein cables to increase the yield of electrical power. So traditionally, we have these unengineered cables produced by Geobacter, which allow, let's say, one electron to move through at a time. However, with our engineered Geobacter cables, allow more electrons to pass through at the same time. In this case, let's say 3 electrons or more, so that more electric power will be generated at the same time. Here is another example from a team of University of Massachusetts who managed to make a prototype of electricity generating devices using these Geobacter cables to continuously direct electrons from air all the way to any external electronic devices. For example, like the LED screen I'm showing you here. In other words, it allows us to harvest electrical energy from air without introducing any pollution. Thus, it brings us a step closer to the dream for green new energy. We can further extend our scope to other bacteria in nature. We have to acknowledge that Geobacter are not the only strange life form with the magic to produce electricity. Nature has us much more. There are groups of bacteria which can produce electricity. Some of them can even form cables together, provide this long-range power supply. Their beautiful surviving stories have been recently studied and revealed by scientists. And I can assure you, soon enough, you shall hear more from these heroic survivors. So today, I hope I deliver enough how amazing our bacteria survivors could be. And I would like to end my part of the talk with New York Times article and remind that there's plenty of strange forms there to be explored. Those strange lives are encouraging you to dream wildly and keep an open mind to any unknown mystery. And of course, next time when you breathe, I hope you'll appreciate more of this magic process. But sit still, because our amazing journey hasn't ended. And I believe my colleague Carrie Ann is going to further mine with more of stories on nature's great survivors, such as water bear. So now I'm going to switch off and switch to Carrie Ann.
All right, thanks Xiangxi for sharing the amazing power of Geobacter. This tiny bacterium, this tiny hero, it's only a single cell, yet its survival strategy of breathing may revolutionize the way we power our societies. So I am Carrie Ann, and in my talk, I'll be moving us up in scale once again from a single cell to an entire organism, an animal in fact, and share with you my favorite survival hero, the water bear. This tiny microscopic hero, seen in the image here, is able to push the pause button on life in order to survive in the most extreme environments. And the coolest thing is that we can learn a lot from them, even from right in our own backyards. So let's pause for a second and think about what is required for life to exist. Well, first, we need to breathe, like our hero Geobacter, and 2, we need water. Water is so fundamental to our understanding of how life arises that space explorations have been driven for the search of water on distant planets. But we already know examples on Earth that can survive without water. Plant seeds are a common example. If you're like me and you've been at home social distancing, maybe you found some seeds in a packet under your cabinet. Who knows how long those seeds have been in there all dried out. But if you plant them and give them some water and some sunlight and some soil, they can spring to life. And I now have some fresh cilantro growing in my windowsill. Bacterial spores are another example of life persisting without water. This is an image of spores from the bacterium Clostridium botulinum. These spores can be found in improperly canned foods and can cause the disease botulism. You could also think of the deadly anthrax spores that were sent through the mail after the 2001 terrorist attacks. And bacterial spores can literally survive for centuries without water. And then they can reactivate when conditions are favorable. One of my favorite examples of life without water comes from a birthday present I received, I think I was around 7, and I got one of these Sea Monkey Ocean Zoo kits. They come with this plastic container and then 3 packets, a water purification packet, some growth medium, and then this packet of instant dried out eggs. And so you mix all the packets with some water, and within 24 hours, I had all of these amazing little shrimp swimming around in the container that came from seemingly nothing. This is a great example of life existing in suspended animation. So the term suspended animation means the temporary slowing or stopping a biological function without death. This may remind you of science fiction stories where humans are placed in suspended states to travel to distant planets to escape dangers on Earth or to explore distant planets. And you can think of these cryopreservation chambers as shown here from this image from the series Lost in Space. You could also think of the films Alien, Avatar, or Interstellar. You can also turn to examples in literature, such as Sleeping Beauty, who enters a suspended state after she pricks her finger and stays in this suspended state until her spell can be broken. We can also think of familiar examples from nature, such as hibernating bears and squirrels, who enter suspended states in the winter when food and water are scarce. And now we'll get to my favorite hero of suspended animation, the tardigrade. So I started talking about suspended animation by talking about space travel, and it turns out that these tardigrades have been able to survive in the vacuum of space, and they may even be on the moon right now. And so in this talk, we'll get to know a little bit about the tardigrade, understand why they're so amazing, how they can enter suspended animation, and what we can learn from them. So first, clearly, tardigrades are not supposed to be on the moon. So where can we normally find them? And it turns out you don't have to go very far. Tardigrades are found nearly everywhere on Earth, even right in our own backyards. So I'm currently living in New Haven, Connecticut, and this is an image of our town green. And New Haven's also often called Elm City because we have so many elm trees. So let's zoom in on this elm tree here, and we'll probably see a splotchy spot. This is an image of a liverwort. And let's continue to zoom in on this splotchy spot and just see really how intricate this liverwort is. And so if we zoom in to the point that we can see the individual cells, if we're lucky, we may also find our hero, the tardigrade. So for reference for scale, these tardigrades are about the size of the thickness of your fingernail. So they're about 0.5 millimeter long. And zooming in on these cells of the liverwort, I'll make them the background of the talk moving forward, since this would be where tardigrades are commonly found walking around. So when I say the term tardigrade, I must be careful to state that this actually is describing a really, really large group of animals. There are over 1,000 species of tardigrades, and they're really adorable. They're really diverse, but I think they're adorable. Just look at the little eye spots and the little claws. You may be familiar with their name, water bear. This comes from the fact that they actually kind of resemble a bear walking around, just that they have too many legs. They have 8 legs. They're also called moss piglets because as we saw on that tree, they're often found on mosses and liverworts on trees around the world. And I grew up in Alabama collecting these off trees with my dad. And one of the coolest places we actually collected them was from the inside of a vacuum cleaner bag. Not sure that made my mom too happy when we busted it open, but this really is just an example of how they're able to survive without water pretty much in any environment. They can survive temperatures from freezing ice to inside volcanoes. And what allows some species of tardigrades to survive these extremes comes from their ability to form a protective state. So if we think about the tardigrade that was on the moss, when it's active, it's going to be full of water, like we're full of water. And then when that moss dehydrates, the tardigrade will also dehydrate it with it and form this protective ton state. This is like a hard cyst-like state. You can sort of think of it like a dried out raisin. And then when the moss rehydrates, the ton state will also rehydrate and go back to the active water bear. So this is a process called anhydrobiosis. Biosis meaning life, and without, and hydro, water. Life without water. So I'll show a video of this here. Here we see the tardigrade in the raisin-like protective ton state, and then water is flown over the tardigrade, and within a couple hours, it's back to its normal daily life, walking around, back rehydrated. And I think of this as similar to how a seed works. If we think of a dried out seed in a packet, it's sort of like a hard protective state, like a ton state. And then with the proper environment, with soil and water and sunlight, it sprouts, reactivates, and then it will grow into the active adult plant. But what is so cool about the tardigrade is that it just doesn't go in one direction from a seed to an adult plant. It actually can go back and forth in this cycle of dehydrating and rehydrating and dehydrating with the moss, as the moss dehydrates and rehydrates with sunny and rainy days. And this protective ton state has allowed tardigrades to survive even in the vacuum of space. So in 2007, a collaboration between US, Russian, and European space agencies launched the LifeTARS mission. So they sent this satellite, the Photon M3, into low Earth orbit for 12 days, and it was carrying tardigrades. And it exposed these tardigrades to the vacuum of space for 12 days. So this includes complete lack of water, in addition to really crazy high levels of solar radiation. So we know that we have to protect our skin from the sun's rays by applying sunscreen to decrease our risk of skin cancer. But it's amazing that these tardigrades were able to survive this really, really intense solar radiation in space for so long. And how exactly they're able to do this is an ongoing area of research. We do know that they were able to survive without water due to this protective ton state. And recently, about a year ago, an Israeli spacecraft was launched to the moon, and its goal was to create sort of like a time capsule of human history. It had some human DNA, some documents about our languages and our music, and it was also carrying hundreds of tardigrades in this protective ton state. However, it crash landed, spilling the tardigrades across the moon. And while we don't know if they survived the crash, if they did, there are currently hundreds of tardigrades in the ton state on the moon. But no need to worry about tardigrades colonizing the moon. They cannot exit this ton state without water, which is just not going to happen on the moon. So unfortunately, they're going to stay there in suspended animation unless they're able to be rescued and brought home. So how exactly can tardigrades enter this ton state? Well, it's an active area of research. But our current understanding comes from the fact that tardigrades must replace the water inside their cells to protect themselves. This is called the water replacement hypothesis. So our cells and tardigrade cells, animal cells, they're basically bags of water. They're chock full of water. They're about 70% water, and this makes for a really happy, plump cell. All the contents inside the cell are surrounded by water, and that's how they like to be. However, if the tardigrade's going to dehydrate, or if a cell is going to dehydrate and lose water, that cell is going to get really shriveled up and unhappy, and everything inside that cell is also going to shrivel up and be unhappy and actually stop functioning. So if tardigrades are going to lose basically all of the water inside their bodies, they're going to need to replace that with something else. And what we know is that they need time to do this. If we plot the survival of a tardigrade from 100% to 0% against the rate at which it is dried out from slow to fast, what we see is that when tardigrades are dried out slowly, nearly all of them survive. However, as the rate of drying is increased, at a certain point, no tardigrades will survive. And while this varies a little bit by species, it is important to note that tardigrades need, if they lose water too fast, they cannot survive. They need time to protect themselves. And work from the University of Chapel Hill actually showed that what they're replacing this water with is actually sticky proteins. And so cells, by making a ton of these sticky proteins, you can sort of think of these as like the headphone cords I'm wearing now. They can get all tangled up and they can be a mess. But they could also become really ordered matrices through a process called vitrification. And when you have a ton of these sticky proteins in the cell, this process of vitrification actually turns the inside of the cell into a glass-like substance. So that's pretty cool, and you can kind of think of this like the Pyrex container, your trusty casserole dish. You can freeze that Pyrex, you can put it in a 500-degree oven, and it is good to go. But if you move from that freezer to the oven too quickly, that Pyrex is going to break. So tardigrades, too, really need time to protect themselves, to build up these sticky proteins, to survive extremes. We saw in the beginning of the talk that while tardigrades are incredible, they're not the only organism on Earth that can survive without water. These sea monkeys I got as a present as a kid also can survive in that packet without any water. And it turns out they're able to do this in a very similar mechanism to tardigrades, but it's a little different. So sea monkey cells also chock full of water. To dehydrate them in that packet, they're going to need to lose that water. And it turns out they actually replace it with a sugar called trehalose. So you can think of this like two glucoses stuck together, and it's pretty similar to the granulated sugar you might be baking with right now. This sugar is able to also, in a process called vitrification, turn the inside of the cell into a glass-like substance so that the sea monkeys could survive without water for a really long time. Now, I love learning about the tardigrades just because they're really adorable and cute and really strange, but it turns out by studying them, we can actually learn about ways that we can improve our society. And so there's currently a lot of research going into using tardigrade proteins, these proteins that are able to protect the tardigrade from losing water, to improve human health. So I want to stress that this is really preliminary ongoing research, and there aren't any applications on the market for this right now. But the thought is that the tardigrade proteins may be able to do things like stabilize vaccines. Right now, the cost of transporting vaccines and getting them to communities that need them across the world, most of that cost is due to having to keep the vaccines cold. So if we could use tardigrade proteins to stabilize the biological substances inside the vaccine, then we wouldn't have to keep them cold, and it would be so much cheaper to get them to the communities that need them. We could also use the same theory to use tardigrade proteins to stabilize traumatic injuries or preserve organs for transplant. So if we could like suspend a human cell in the midst of a trauma in place so that the damage doesn't get worse, we would have more time to get someone after suffering a traumatic injury to an area that could help them or to preserve that organ from the donor to the recipient for transplant. So right now, where we are in the process is that tardigrade proteins can stabilize bacteria and fungi cells against drying out, but there is quite a lot of work to be done before these applications are realized. But keep your ear out. I do think there'll be some promising research coming from this in the next few years. So that has been our story of the hero, the tardigrade. We have seen that they are these adorable, cartoonish little water bears that are able to survive extremes due to this protective raisin-like tongue state. And they're found nearly everywhere on Earth, but most importantly, right in our own backyard. They're able to survive extremes by replacing the water in their cells with sticky proteins, which through a process called vitrification turn the inside of the cell into a glass-like substance. And finally, by learning about how our strange survival hero can survive such extremes, we may be able to rethink how we stabilize vaccines and preserve organs for transplant. So in tonight's talk, we've now met 3 very different survival heroes that span from the Autophagosome, cleaning up the trash inside of our cells and protecting us from starvation, to the single-celled Geobacter, capable of producing electricity from this simple process of breathing. And now we have met our animal friend, the tardigrade, that is able to survive without the most basic of life-giving substances, water. So each of these heroes has a very different type of superpower at very different scales. But together, they can really teach us just how crazy nature can be. So with that, I would like to conclude tonight's talk by thanking all of our Science in the News speakers during this 2020 season. We've had a lot of great talks and are really grateful to have been able to adapt to the online format and bring you our talk tonight via Zoom. And we look forward to coming back in 2021 with a new series of talks and hopefully in person.